All right, so let's start, give you a little bit. We were told the topic here. So the background is that obviously extracellular vesicles have become a very important part of my life, and they're a huge kind of component in this whole cell function, disease state, and communication of cells. So, and they've really lended themselves directly to being a prognostic and a diagnostic indicator. And so what we want to do is move on where most groups have and look at what's inside, right? It's like us, it's most important is what's inside. And so we want to look at these microRNA levels. This seems to be the central point for looking and using them in kind of, using biofluids and whatnot to actually do some sort of diagnosis or prognosis on an individual. So we started off and thought about how should we actually go about doing this. So, won't advance. There we go. So microRNAs can be looked at in many different ways. You can use RNA-seqs and PCRs and Western blots and all kinds of genomics and stuff like that. Your omics are a huge portion of it. But what we started thinking about is these are not kind of systems relative, right? So they're not natural to us or how things, the biology would work inside of the human system. And so the other things we started to think about too was, okay, well, you have these difficult extraction protocols, you need lots of amplification. Your expression level is kind of biased towards if you have to have a higher level, hence the amplification steps. So we said, how can we do this and actually, do it more efficiently? So we started to look at these things called molecular beacons, and they offer us a method of looking at a specific microRNA in the biofluid. And we wanted to actually complement this to our normal works of nanoflow cytometry for extracellular vesicles. So what we did is take these molecular beacons. This was something done by a group in Rutgers, so hence the little MetroFlow tie there. And it was Tiagi, and back in, God, I think his first publication on them was back in, say, 94. And it's basically a stem loop sequence where you have a reporter and a quencher. So you have a fluorescence tag along with like what they call a black hole quencher. When they're in close proximity, you will not see fluorescence. Once you get in a target, the stem loop system opens up and it becomes fluorescent. In our studies, we look at red blood cells, the EVs derive from them, and hence we're going to be looking at specifically in this instance, microRNA 451A, which is, inside of the red blood cell EV. The method's pretty straightforward and simple. You take your molecular beacons, you add them to your cell of interest, and with the microRNAs inside, you let them incubate, mix them up. These guys penetrate in, and when they find a target sequence, they unloop, and then you get your detection using the nanoflow cytometry. The big thing here is you're going to see this little CPP lettering, and basically a CPP is a cell penetrating peptide. That's going to bind onto these molecular beacons with the sequence, the oligonucleotide sequence, and it's actually can penetrate the cell membrane or the membrane of the EV and allow for this interaction to happen. Without the CPP, they just stare at each other. All right, so we wanted to see it in the actual red blood cells. So we took it, we labeled those with a cell mass green. We used an Alexa 594 on the molecular beacon. You can get the beacon on the fluorescence and basically anything. You just tell the company, hey, this is what I want, and they add it. So we have differing levels here. We used a FAM, we used an Alexa 594. At one point, there was a APC derivative. So the point of this slide is to see that, okay, we can do it in the cell. Also to show off that we could actually do super res microscopy. So that was a big thing. And basically then show these guys inside and even some where the membrane starts to bud. The two bottom pictures are actually showing these inside the extracellular vesicles, the exosomes here. And this was done by just taking the red blood cells, sprinkling them with, say, an ionomycin or an A23187, and they generate a ton of these EVs. And when they bleb off, they send them along with their microRNAs. So this gave us the ability to see how the system worked. So we were using a Cytoflex S, and basically, we could see things doing what we're calling nanoflow cytometry, nanofax, whatever other wording that you've heard. And then what came to right was this thing called a Mustang, now known as the Cytoflex SRT. We had the ability to get one kind of pre-manufacturing and said, well, if our Cytoflex S is able to do this, then this supposed Cytoflex that sorts should also be able to do it. But you can't just go in blind. So we did some testing. We had a group here at Northeastern University that gave us some bacteria. Beads are a dime a dozen. We can get those. I believe we grabbed them from Thermo Fisher. And then we had these virus-like particles from a collaborator at Boston University. So we're going to do a little bit of testing. And because we are who we are, we decided, hey, let's do single deposition because that's much easier than doing bulk sorting. And so we decided here to start with our Bacillus subtilis and sort a single one of them. So that's what that big kind of green circle is. Should have done a rod. And there you can see it underneath the fluorescent microscope. And so we actually kind of did it on the first try and surprised ourselves slightly. But then said, okay, let's get a little more challenging. Let's take those beads. We're going to mix two different ones together, 100 and 150 nanometer size. We'll start by doing a lovely 5-spot, and then we'll do 1. We had a target, if you notice, we missed, which I appreciate Rui also showing his animation, how it missed. So that was a good setup. Thank you, Rui. It was still in the field of view on the microscope, so that was great because otherwise we would have thought we failed. Don't really have an explanation except for maybe some sort of static charge there. So then we went to these virus-like particles and we sorted the fluorescent positive. This is a GFP GAG polyprotein. And we used an M-Cherry variant of the same line to set up what would be our negative gating. And then we just sorted GFP positive, GFP negatives. All right, so now we're down to something, and this particular virus-like particle has a size range between 90 and 130 nanometers. So we went to our actual molecular beacons, and with the microRNA 451, these are the red blood cell derived EVs using the A23187. And so we set this up with just a control EV, one that did not have any molecular beacons. We did that and compared it to the ones with the molecular beacons. We used a fluorescence trigger here. That's what's on the top where it says FL, simply because of what we wanted to try to prove is that possibly things that didn't register themselves as fluorescent positive that would be in that negative gate may still be fluorescence. It's just the sensitivity of the instrument's not great enough to push them past the particular line we were setting. So here that's showing that once we actually sorted them, the red is the actual violet side scatter proportioning of the PITC here from the molecular beacon. It's a FAM actually, so it's a FITC derivative. And the bar is showing you how in the violet side scatter, that would be the stuff that would be considered absolutely not a vesicle. And everything else was kind of positive. Using FCM pass, we were able to see the two different populations, the one that would be considered low or negative fluorescence, showing 500 MESF values, and comparing that to the high fluorescence of 2500 MESF values. And once they were sorted, these two particular populations that are the blue and greens, we then took them and did some PCR on them because we just didn't want to say, oh, good, we could sort them. We wanted to see what we actually had. So we took 4 different people. We sorted about 400,000 events of each of those blues and greens there on the histogram, did the CT value, so the cycle threshold, and found that the average was exactly the same. The numbers are pretty close. If you graph them out and look at them, obviously we just did it this way for brevity. And it surprised us that we did have the fluorescence and the microRNA sitting in the population that the instrument would have called negative. So here's why it all matters to us. Because when we started doing and looking at the TEM, we were able to see that there were small EVs or more exosomes, maybe in exomeres, and other large EVs or the microvesicle populations. And you can see that as they go along, whether it's in the flow through the TEM or using some sort of resistance pulse sensing, they were all showing this multiple population, these small and large, instead of just one bulk population. It split them pretty well into two homogeneous populations instead of one heterogeneous kind of splurge. So basically what we saw there is that doing the flow, we were missing a lot of stuff. It's those things where they show you the tip of the iceberg. Well, we're starting to dig underneath the tip and see things underneath what that iceberg is actually formed as. Maybe the Titanic would have liked this technology here. And then we looked at them again with microscopy showing, yes, there's more of the large ones. They do show up brighter and you can see the detailings there, but the small population exists and we can pull it out. And thanks to sorting, we were actually able to do that. Hopefully I talked fast enough. All the acknowledgements of the people that were instrumental in helping us figure this out and kind of understand some of the things that we were looking at. I put this reference page up just to make it a little more, so if someone wanted to take a quick picture of this, or if it's available to you, there's some papers there that would explain better what we did. It's obviously in a very quick 10-minute process, not that easy to go through. There was a lot of things missing. So I tried to get the details across without, skipping too much of the meat of the sandwich. And then we just can go down to, if you have questions, I know we have the group thing. I don't know if I talked fast enough to give myself any time right here, but there, I hope was a decent presentation. Thank you.
That was absolutely wonderful presentation, John. And we do have about 3 minutes left for questions. So we do
Nailed
have
it.
a couple. Yes, that was absolutely perfect. Couldn't ask for better. The first question that we actually have is coming from Steve McClellan, and he's asking if the 100 micron nozzle is a limit to the volume you get back, and is it more difficult to recover the sorted EVs with that 100 micron nozzle? And he also said thank you for the talk.
So Steve, that's something that gets missed on this brief one is the volume actually out of an SRT. We did some comparisons, and off a 100 micron nozzle tip, because of the decreased pressure, is what my thought is, you're actually getting less volume than you would with the Aria or the Astrios, or if you have an XDP or anything like that. Secondarily is we're actually able to concentrate the samples. We just use Amicon filters, take all the sorted sample down and put it on there, and then the fluid comes out and we get a concentrated sample. So that's what was actually being tested in the PCRs.
Awesome. I also have another question. So when you were testing out the APC conjugated or the APC that was in the molecular probes or the molecular beacons rather, did you find that the molecular weight of APC caused any issues since it's a larger molecule in comparison with some of the Alexa dyes?
We didn't really have that problem of the molecular weight because we kind of cheat the system in a little bit of, we don't just go after the EVs originally. We, as stated, label the cell of interest and then cause them to vesiculate and they pull in. So they're already kind of labeled and
Gotcha.
then we're taking them. So it's kind of like they're already inside. We've done it where you can just do it and release, you have to do the check. So we did it with just releasing the RNAs. And some of that was simply why they kind of twofold. One is the FAM is less, it's smaller and whatnot. Two is for some reason it's less expensive when you order it. So twofold why us in academia went with this wonderful. I'd like to say it was completely science-based and what you just said. Most of it was based on, wait, we can save a couple of $100.
Yep, that completely makes sense. And work in a core, or anyone that works in a core can understand that, especially in academia now.